Okay, so this lecture will be about cells and transport. Everything in this lecture will link to the cells and transport topic from the AQA Biology Paper 1 specification. This is mainly directed at combined science students, though is useful for triple science students also. So the key rules for this session: make sure you've printed out the booklet from the link below the video. If you've got the booklet, you don't need it in stage 1, but you will need it in stage 2 and stage 3. So, what is spatial learning science? So the plan is, in stage 1, I'll talk, you'll listen and watch. You don't write anything down, you just have to listen. You're then going to go and take a 5-minute break. You're then going to go back to the start of the video and you're going to repeat it. Once you repeat it, I'm going to talk, but you're going to do simple tasks. You're going to fill in the gaps and complete simple calculations. You'll then take another 5-minute break, and then in stage 3, you'll complete harder tasks. Now, the answers will be available at the end, but these are sort of more like the questions that you'll face in the exam papers. So we're slowly making it more complex, from you just listening, to you filling in the gaps, to you answering questions. So here we go, stage 1. I talk, you listen. Put your pens down, eyes to the front, watch the video. Here we go. Okay, types of cells. Plant and animal cells are eukaryotic cells. They have a cell membrane, cytoplasm, and a nucleus. Bacterial cells, prokaryotic cells, are much smaller in comparison. They do not have a nucleus. So eukaryotes, yes, they have a nucleus, yes. Prokaryotes, no nucleus, no. So eukaryote, yes, nucleus. Prokaryote, pro rhymes with no nucleus. Observing cells. To see cells, we need to use a microscope as they're so small. We now have electron microscopes, which have allowed us to see what is inside cells. So we have light microscopes and electron microscopes. Now, electron microscopes have a much higher magnification and resolution. Magnification is the amount we can zoom in on an object. The resolution is the ability to see the difference between small objects. So 2 key definitions: magnification, amount we can zoom in on an object; resolution, the ability to see the difference between small objects. You have to be able to calculate the magnification, and you should be able to calculate magnification of an image or object using this equation. An easiest way to remember it is I am in a formula triangle. I stands for image size, A stands for actual size, and M stands for magnification. So if you want to work out any of these, you can cover the thing you want to work out, and it tells you the sum. So if you cover I, it's A times M. If you cover M, it's I divided by A. I is image size, A is actual size, M is magnification. So let's have a look at an example. Worked example 1. A magnified animal cell has a diameter of 6 millimeters. The actual diameter of the structure is 0.15 millimeters. Calculate how many times the structure has been magnified. So 6 millimeters, that's the image. It's 6 millimeters in size. The object is 0.15 millimeters. So to calculate magnification, I do I divided by A. So magnification is equal to 6 divided by 0.15. So the magnification is 40. Now you may need to write your answers in standard form in the exam. So, animal cells are eukaryotes. Remember, yes, it has a nucleus. You need to be able to label an animal cell. So on the outside there, you've got the cell membrane. That controls what enters and leaves the cell. You've then got a ribosome. Ribosomes are involved in the process of protein synthesis. You've got a nucleus, which stores all of the DNA. You've got cytoplasm, which is where chemical reactions take place in the cell. And you've got mitochondria. Mitochondria are the site where respiration takes place. So energy is generated. You then need to be able to label a plant cell. A plant cell, again, is a eukaryote. Yes, eukaryotes have a nucleus. Now, the plant cell is very similar to an animal cell. However, it also has a cell wall, which helps to keep the shape. It's got a cell membrane, which controls what enters and leaves. It's got ribosomes for protein synthesis, nucleus, which contains the DNA. It's got a vacuole, and a vacuole contains cell sap to decide whether to give the cell its turgidity. A cytoplasm, where chemical reactions take place. Mitochondria, where energy is generated via respiration. And then they've also got these green chloroplasts. These are the site where photosynthesis takes place. So you need to also be able to label a bacterial cell. A bacterial cell is a prokaryote. No. Prokaryote, no nucleus. Now the outside of this one has a cell wall. They've also got flagella at the back, and these flagella can be used to help with movement. They've got a cell membrane which controls what enters and leaves, but then they've got two types of DNA. Chromosomal DNA and then a simple ring of DNA which is called a plasmid. A plasmid is really important in one of our future topics, which is genetic engineering. So, what are the functions of the cell parts? Both animal and plants have a nucleus which controls the cell and contains chromosomes, the DNA. They have a cytoplasm where chemical reactions take place, cell membrane which controls what enters and leaves the cell, mitochondria which generates energy by respiration, and ribosomes. These carry out protein synthesis. They make proteins. And we've then got the parts that are just in plant cells. So the chloroplast is a site where photosynthesis takes place. The vacuole is filled with cell sap, and the cell wall is made of cellulose, and it strengthens the cell. Now you also need to know about cell specialization. You need to know how cells are specialized to carry out their jobs. And I'm going to go through some key specialized cells over the next few slides. So the first one's a sperm cell. Sperm cell is used to fertilize the egg in reproduction. And it has a streamlined head and tail to help it swim. It also contains lots of mitochondria to give it lots of energy. The next one's a nerve cell. These carry electrical signals around the body. It's got a long structure to increase its distance, and it's got branched connections to join to other nerves. So these dendrites are the branched connections. We've got a muscle cell. These contract and relax to bring about movement. They've got lots of mitochondria to provide energy, and they're long, giving them space to contract. From plants, we've got a root hair cell. Don't get confused and think that this is a root hair in your head. This is a plant, this is part of a plant, it's part of a plant's roots. So the root hair cell absorbs water and minerals from soil. And to help them do that, they've got a large surface area to increase absorption. We've also got xylem. These transport water and minerals around the plant. And these have got hollow tubes. There's phloem, which transport glucose around the plant. This is called translocation. Now you can see from this diagram that the cells at the end each have holes at the end so that the glucose solution can move through. Now separate to cell specialization, you also need to know about cell differentiation. Now, differentiation is when cells change to become different. The clue is in the name. Differentiation, different. Now, some cells can change to form different types of cells. Now, in animals, this mostly happens when we're young. Adults only rarely repair and replace cells, but they don't do a great deal of that. Whereas plants happens throughout their life. So through a whole plant's life, they'll continually differentiate their cells. Now, a key part of that cell differentiation is the cell cycle. So cells need to divide in the cell cycle for growth and repair. Now, this process is called mitosis, and we're going to look at mitosis in more detail. Now, before we look at mitosis, it's important that you know about chromosomes. Now, the nucleus contains chromosomes made of DNA. Each chromosome carries a large number of genes, and in body cells, the chromosomes are normally found in pairs. So, what happens in mitosis? Mitosis involves cell division to produce two identical cells. In stage 1, before the cell divides, it grows and increases the number of ribosomes, mitochondria, and makes two copies of every chromosome. Now, in the second stage of mitosis, one set of each chromosome is pulled to each end of the cell, and the nucleus divides. So we doubled the number of chromosomes. We now pull one of each of those to each end, and the nucleus divides. And in stage 3, the cytoplasm and cell membrane will divide to form two identical cells. You can think of mitosis as mitosis. Two identical cells are produced. And here's that process shown in one diagram. So the mother cell replicates, so all of those mitochondria, ribosome and chromosomes are doubled. They then get pulled to each end and the nucleus divides. And then the cytoplasm divides and we end up with two identical daughter cells. And on to the last part, stem cells. A stem cell is an undifferentiated cell. Undifferentiated means it's not yet changed to a different type. These cells can change and divide into other cells and cells of the same type. Now, there are three types of stem cells you need to know about. Embryonic stem cells can differentiate into any type of cell. Adult stem cells can change into less type of cells. But then in plants, we've got the meristem tissue in plants. These can change into any type of cell for the plant's whole life. Now, stem cells can be used for lots of things. Therapeutic cloning can produce embryos with the same genes as a person. These can then be used to treat disease. Stem cells will then not be rejected by the person's body. Some people think it's bad though, as it damages an embryo and goes against people's religious beliefs. In plants, they can also be used to produce clones. So it stops rare species going extinct, and you can also produce more plants which are resistant to diseases. So if you've got a particular plant that's resistant to a disease, you can clone that using the stem cells. Right, that's the end of stage 1. Now all you need to do now is you need to go and take a break for 5 minutes and forget about that information. Then when you come back to start stage 2, I want you to skip to the start of the video and replay the first part of the lecture. Now, as you listen to it this time, you're going to complete the close activity. You're going to fill in the gaps on the booklet for stage 2. So make sure you've got that booklet and fill in the gaps for stage 2. Now, hopefully, you've just completed stage 2. That means you're now ready to go on to stage 3. Again, you need to take a break, a minimum of 5 minutes. Now, in stage 3, you're going to press pause at this point. Once you've pressed pause, you're going to answer the questions in stage 3 of the booklet, and you've got 15 minutes to do that. When you press play, the answers will then appear over the next few minutes. Right then, let's go through the answers. So what is an eukaryote, a eukaryotic cell? A eukaryotic cell is a cell that has a nucleus. Eukaryotic, yes, it has a nucleus. What is a prokaryotic cell? No nucleus. Which microscope is more powerful? An electron microscope. Which one has a bigger resolution? An electron microscope. What is the equation to calculate image size using actual size and magnification? Image equals actual times magnification. And if the actual size is 1 millimeter and the magnification is 20, how big is the image? So you do 1 times 20, 20 millimeters. So what type of cell is this? It's the one on the right hand side. That is a plant cell. What is part X? Part X is the nucleus. And what does X do? X controls the cell. Now label the bacterial cell. Hopefully you've put cell wall and plasmid DNA. What is the function of chloroplasts? Chloroplasts are used in photosynthesis. As long as you write photosynthesis, you will get a mark. What's the function of mitochondria? They generate energy. And again, you should try to include the word respiration because that's the process. Which part carries out protein synthesis? Protein synthesis is carried out by the ribosomes. What does a sperm cell do and how is it adapted? The sperm cell fertilizes the egg. It has a streamlined head, tail, and lots of mitochondria. What transports water and minerals around a plant? That's the xylem. What transports glucose? That's the phloem. And why are root hair cells important? They absorb water and minerals. What is meant by cell differentiation? That's when cells change to become different. Why do cells need to divide? Well, it's because they repair and growth. And what are chromosomes made of? Chromosomes are made of DNA. Right, you should now complete this passage about mitosis. So stage 1, before the cell divides, it grows and increases the number of chromosomes, number of ribosomes, mitochondria, and makes 2 copies of every chromosome. Stage 2, one set of each chromosome is pulled to each end of the cell, and the nucleus divides. Stage 3, finally, the cytoplasm and cell membrane divides to form 2 identical cells. And finally, what are stem cells? They are undifferentiated cells. Name the 3 types of stem cell: embryonic, adult stem cells, and meristem cells. Which type of human stem cells can change into most types? Embryonic. What are the uses of stem cells? They can be used to treat diseases. And what are the advantages of using therapeutic cloning? The patient's body will not reject the cells.